When I told David last week that I was introducing him, he had some advice. He said, keep it brief. So here we go. This was an assignment not easily met when the speaker is one of the foremost fungal biologists and evolutionary biologists in the country. He grew up locally, education at UMass Amherst and Duke, where he got his PhD, postdocs in Japan, and luckily for us, here. Now professor at Clark University and fellow this year at the Radcliffe Institute. He has worked at many levels. It's one of the things that's most interesting about David's work. Systematics, a little bit of nomenclature even, phylogeny, biogeography, genetics and development, genomics. In short, he has worked from shiitake to fungi trapped in amber to coal deposits. His project at Radcliffe focuses on a book about phylogenetics of fungi presented for the general audience. He has had many students and postdocs who have worked in these broad areas that I've outlined for David. So this is the short version. Here's the long version and the real version.
Well, thank you very much, Don, for the nice introduction, and Jane for the introduction. Thank you all for coming. It's really amazing and wonderful to see such a great crowd tonight. I suppose I should also thank the donors who have supported this series, and I certainly want to thank the Radcliffe Institute for giving me this really wonderful year at Harvard. It's been a thrill. So I'm a fungal biologist, I'm a fungal evolutionary biologist, and I'm really excited to have this chance to tell you a little bit about my research. But before I can do that, there are a couple of nagging questions that I feel compelled to dispense with. And the first one is, since this is a microbial seminar series, the question is, are fungi microorganisms? So I need to make sure that I'm in the right place and that you're in the right place. So depending on how I answer this, you can let me know if I can stay or not. So are fungi microorganisms? Well, obviously the answer is yes, because we have things like yeasts and molds, which, well, when you get many, many, many of them together in one place, you can see them with your eye, but individually, clearly too small to see. So are fungi microorganisms? The answer is clearly no, because there are also clearly some macro fungi, like this magnificent polypore decaying this beech tree. So that's about as macro as an organism gets. So it's not an easy question to answer because there is no one answer, right? So it's a trick question or a false dichotomy. So fungi, mushroom-forming fungi that I work on have both micro and macro phases. So the mushrooms that you see growing on a tree trunk or out of the ground are clearly a macro phase. They have one function in life, and that is to make spores, which are microscopic individually. The spores germinate to form these microscopic threads, which collectively form something called the mycelium, which you can see with your eye. But usually you won't see that because it's down in the substrate, which could be soil or a rotten log or a loaf of bread in your kitchen or your toenail if you're so unfortunate. But the mycelium is the phase of the fungal life cycle where really all the action happens, because this is the phase of the life cycle where the organism explores its environment and captures territory and feeds and fights with other individuals of its own or other species, mates. And when conditions are good, this mycelium will push up a mushroom. Okay? So this mycelial life cycle makes fungi really hard to study because most of the time when they're doing lots of interesting things, they're out of sight in the substrate. And this has led to some real surprises. Some of the biggest surprises come from the genus Armillaria, which I think some of you have probably heard about. This is the genus that includes the so-called humongous fungus, right? So Armillaria makes these little mushrooms that pop up in the forest floor. The mycelium has a neat trick. It makes what are called rhizomorphs, which are root-like aggregations of hyphae, which you can see in the Petri plate on the right. And it uses these to explore the forest floor. And mycologists have known for ages that these rhizomorphs could potentially link fairly large clones. But it was only when we used genetic methods that we could get a sense of how big some of these clones really are. And the biggest one that I know of, and maybe that mycologists know of, is a fungus that's out in Oregon in the Malheur Forest. They call it Genet D, which, if you ask me, is not a very romantic name for such an interesting organism. I think they should give it an individual name so we could maybe suggest something. Anyway, when people have used these genetic techniques to assess the extent of an individual, Genet D turns out to be 2,300 acres in area, at least 2,000 years old, and at least 7,500 tons. So think about that, right? So Genet D is one of the oldest and largest living organisms that we know of on the planet. Now, you can quibble about whether it's an individual or a clone, but nonetheless, it is one big fungus. And it turns out, coincidentally, that Genet D, the area of Genet D, is almost exactly the same size as Logan Airport. But Genet D is a lot older than Logan Airport, and it may be around a lot longer after Logan is gone. In any case, the next time that you're taxiing out on the runway at Logan, look around and realize that all of what you see around you could be covered by one single fungus. This illustration, by the way, is by a former student of mine, Amy Yeager. I don't know if she's here, but this is for an exhibit on fungal biology that's going to be coming along at the Ecotarium in Worcester later this summer. Okay, so are fungi microbes or macrobes? The answer is both. Now you don't trust me because I've misled you with that. The other question that I want to get out of the way right away here is, why would anybody study mushrooms for a living? And I know that little boys and girls do not grow up dreaming of becoming a mycologist someday, at least not most little boys and girls, maybe some, but most don't. And I know this because I deal in my work at Clark with first-year college students. And you know, when I tell them that I work on fungi, they give me funny looks. And my colleagues who work on fruit flies and sticklebacks, they don't get that. So I have a bit of a chip on my shoulder about this. And I have evidence of what the students think. There's a website that academics can go to called ratemyprofessor.com. It's completely unedited. And and we all go and check it out. And so here are a couple quotes. So this first person said some nice things about me, very gratifying, but then said, "Too bad he studies mushrooms for a living." Okay? This is real. And the second person said, "Surprisingly not boring for a mycologist." So, ouch. Anyway, I feel like, given this audience, I don't need to tell you why it's worthwhile to study mushrooms for a living, but I feel like compelled to say a few words on behalf of mycology anyway. So why do fungi matter? Well, one reason is that some of them are edible, right? So some mushrooms are edible, or as my daughter once pointed out, any mushroom can be edible. You can eat any mushroom once. But these are mushrooms you might want to eat more than once. They include some wild things like porcinis and domesticated things like shiitake. So mushrooms are edible, some. Fungi are agents of fermentation. I think you all know this, but without yeast, which is a fungus, we would have no beer, no wine, no bread. What a drab existence that would be. The same fungus is used to make biofuel, so the ethanol in biofuels is prepared by fermentation by fungi. And fungi are sources of all kinds of beneficial biochemicals like antibiotics. Most of you probably know that penicillin, the first antibiotic, was discovered in a mold, Penicillium, and lots of other drugs like statins, for example. So fungi impact your lives in positive ways every single day. But fungi also have they have a dark side because fungi cause disease. And some of the diseases that fungi cause are diseases of plants. And there are innumerable fungal plant diseases I could have picked. The one I'm showing here is chestnut blight. So the eastern deciduous forest used to be dominated by these magnificent chestnut trees with these beautiful straight-grained dense timbers. Doubtless the building that we're in has chestnut timbers in it. Anyway, a fungus came along and wiped them out. So the chestnut trees are reduced to just little stump-sprouting, pathetic things in the forest now. So fungi cause very important plant diseases, wheat rust, et cetera. Fungi also cause diseases of animals. and we are animals. This is the only gross out slide in the talk, you'll be happy to know. So we are animals, so we suffer from fungal diseases. Some are trivial annoyances like toenail fungus and ringworm, which seem to be a great obsession these days. This is a fungal disease, not really terribly serious. But some fungi can cause really serious diseases. This is an obscure plant pathogenic fungus that can opportunistically cause infections of people. And in 2012, there's a very terrible story of local interest in which a compounding pharmacy not far from here in Framingham, because of bad lab practices, had a batch of injected steroid drugs that became contaminated with this fungus, caused terrible spinal meningitis. 76 people were killed, hundreds were sickened, and thousands were worried sick, as the headline says, because they'd been exposed to this fungus that normally is not a big problem for people. And the president of the company was just sentenced last year to 9 years in prison. So this is another reason why fungi matter. On a happier, more positive note, one of the reasons that I like to study fungi is because there are just so many of them, and so many are undescribed. Now, there are about 135,000 described species of fungi to which we've given scientific names. That's a big number. But the number of extant species is really just, it's a matter of conjecture, right? So 1.5 million, 5 million, those are common estimates. But there are recent serious estimates that go from 600,000 to 650 million. These are serious published estimates. So we don't know to within several orders of magnitude how many species of fungi are really on the planet. So there's a lot of discovery to be made. Now, most of these undescribed fungi are probably molds and yeasts and other micro things. Probably not so many macrofungi, but there are lots of macrofungi, surely lots of macrofungal species to describe yet. And if you're interested in getting to know the local macrofungi, I want to give a shout out here to the Boston Mycological Club, which is the nation's old, the nation's oldest amateur mycological society based here at Harvard. It's a wonderful organization. So those of you who want to learn some more about our local fungi, you should check out the BMC. Okay, so with that general introduction to what fungi are and why you should study them, let me tell you about some of my work. So the group of fungi that I'm most interested in is called the Agaricomycetes. It's in part of a slightly more inclusive group called the Agaricomycetina. And that name might not be real familiar to you, but you all know these things because these are the mushroom-forming fungi. So the vast majority of fleshy fungi that you see out in the woods, polypores, coral fungi, guild fungi, they're in this group. They're beautiful. And there are roughly 25,000 described species. So it's a fairly large branch of the fungal tree of life. And I'm interested really in mainly two there's people over there too. Two aspects of their biology really fascinate me. And one is their morphological diversity. And this is really what drew me into working with these things in the first place, because they're beautiful and they're wildly diverse. I mean, you've got coral fungi, shelf fungi, stinkhorns, et cetera. So I've been very interested in understanding the patterns of evolutionary changes between these morphologies and whether there are any general trends. But that's not what I'm going to talk about today. Instead, today, I'm going to talk about ecological diversity, because the morphological diversity of Agaricomycetes, I would say, is matched by the diversity of strategies that they use to obtain nutrition in nature. And here I'm talking about carbon nutrition, where they get the building blocks for all of their organic molecules. So we can divide Agaricomycetes nutritional modes into two broad categories: saprotrophs, which are the decayers that attack dead organic matter, and biotrophs, which don't wait for the organic matter to die. They partner with other living organisms, or they cause disease or whatever. We're going to focus today on wood decay, mainly on wood decay, of which there are two types called brown rot and white rot, which we'll introduce in a moment. And we'll also briefly touch on ectomycorrhizae, which are symbioses between plants and fungi in which there's an exchange of sugars coming from the plant to the fungus, which is reciprocating by helping the plant obtain mineral nutrients. So today we're going to focus really on wood decay. So I think it's appropriate to say a few words about wood. Wood is an amazing material, right? So the evolution of wood was one of the great innovations in the history of life, I would argue. It's hard to imagine our lives without wood. It's this beautiful, durable, rigid, useful material. We make utilitarian objects from it, boats, musical instruments, and we build buildings out of it. Rarely as magnificent as this building, but we use wood all the time to build our homes and our podia. Okay, so it's this amazing, useful, durable, rigid stuff that generally is pretty resistant to decay. But what is it? So wood is plant cell walls. They're secondarily thickened plant cell walls, and it's not a homogeneous material. It's actually very complex. And there are a lot of components to wood. The major components are illustrated here. So on the top in green is cellulose. This is a carbohydrate. It's made up of sugar subunits, repeating subunits. Cellulosic ethanol is one of the great holy grails of the biofuel industry. If we could figure out an efficient way to pull off those sugars out of cellulose and use them to make fuel, it would be fantastic. Hemicellulose is a branched carbohydrate, so on the lower left in blue. So hemicelluloses are also made of sugars, but they're branched polymers instead of linear like cellulose. So cellulose and hemicellulose are the goodies that the fungi are after. This is what they're using to get energy and to get the carbon that they're going to use to make all of their own organic compounds. But to get, and also cellulose is what we're after when we do things like biopulping to make, pulping to make paper and so on. But to get to the goodies, to get to cellulose, the fungi have to deal with the compound on the lower right in brown, which is lignin. Lignin is a heterogeneous polymer of these sort of alcohol subunits. They're attached in all kinds of crazy ways. It's tough, it's rigid, it's resistant to decay, and it surrounds the cellulose and protects it from microbial attack. And it's the #1 barrier to cellulosic biofuels. So wood is complex, and there are also proteins, various extractive compounds, et cetera, but these are the major compounds. So wood is complicated stuff, and fungi, to attack wood, they have diverse enzymes that they use to attack different components of the wood, and they work in concert. This slide here, which is, I know it's rather busy, it lists a bunch of the important enzymes in wood decay. And I'm going to talk in this today about the ones that are in the white boxes. So class 2 peroxidases, which attack lignin, and then these others that attack cellulose that I'll speak more about later. So enzymes are proteins. Proteins are encoded in genes, in DNA. So the genomes of fungi have lots and lots of gene copies for these various enzymes that are used to attack wood. So enzymes, I am no enzymologist, but I have friends who are, and they have convinced me that enzymes are amazing. Not that I needed any convincing. This is one hell of an enzyme here. This is called a cellobiohydrolase, GH6 in the previous slide. This is a cellulose-degrading enzyme. The green is cellulose. That's a ribbon model of the enzyme itself. It's got the carbohydrate binding module, the CBM, helps it adhere to the cellulose. This thing crawls along the cellulose fibril, peeling off the cellulose polymer and clipping off little subunits of just 2 sugar molecules that other enzymes can then attack. This is a very sophisticated molecular machine for attacking cellulose. So this is one example. Okay, so wood decaying agaricomycetes have 2 major mechanisms for doing wood decay, which are called white rot and brown rot. White rot is illustrated on the left. The substrate, the wood, is left white and stringy after colonization. Brown rot on the right, it's brown. It kind of breaks into these cubical chunks. In white rot, all the components of the plant cell wall are degraded, including this very recalcitrant lignin fraction. And in brown rot, so white rot is kind of the brute force method of decay. Brown rot is so sophisticated. In brown rot, the fungi have figured out a way to loosen up the plant cell wall with very reactive small molecules, hydroxyl radical. They just sort of loosen up the cell wall, and they can start to pull out the carbohydrates while leaving the lignin behind. So that brown material is almost all modified lignin. And I think we have some samples of white rot and brown rot. somewhere that can be passed around. So those will be distributed. There'll be a couple trays with some rotten wood. If you think you get funny looks when you're collecting mushrooms, think about the looks you get when you're collecting rotten wood. But they're dry, they're not going to harm you, and you can pass them around. And I encourage you to crumble the stuff up in your fingers, and you can see the difference in texture, and realize that this is coming from the same starting material, which is this tough, rigid material that we build our houses out of. Okay, so that'll be going around. And go ahead and break it up. Okay, so white rot and brown rot. And in my lab, one of my major endeavors has been to understand evolutionary transitions between these decay modes, between brown rot and white rot, and between saprotrophy and ectomycorrhizal symbiosis. And this turns out to be a really hard problem. So this is the first of many phylogenetic trees I'm going to show you tonight. This is an evolutionary tree. It's a model. It's a hypothesis of relationships. The branching points on the tree where the lines diverge are speciation events. That's how the tree of life grows, by branching and speciating. And on the tips of this tree are groups of fungi. So this is an organismal phylogeny. And I'm going to minimize use of these taxonomic names tonight because they're really not important for our purposes. So this is an organism phylogeny. And again, the nodes are speciation points. Later, I'll show you a phylogeny where the things at the tips are not species, but genes. And you can see, WR is white rot, BR is brown rot, ECM is mycorrhizal. And you can see that the white rot things are not all in one branch and the brown rot things in another, but rather these nutritional modes are all mixed up across the tree, the fungal tree of life. So there have been lots of transitions in different directions, and reconstructing this pattern is very hard. And understanding its genetic basis is yet another challenge. Okay, so this is a major endeavor from my lab. Now, at this moment, I have to say a couple words about coal. Coal is a mega commodity. It's the remains of undecayed plant matter, as I think you know. Billions of tons of coal are still harvested or mined every year. Huge volumes of coal are recoverable yet. And we're still getting about 30% of our electricity in the United States by burning coal. That's gone down in recent years. Some people would like to see that go up, but it's been going down, thankfully. So the brown field in the graph on the bottom, that bottom light brown, is coal usage as an electricity source in the United States. So coal is amazing stuff. Well, where did it come from? So most coal deposits, not all, but most were formed during a relatively brief interval of about 60 million years, the Carboniferous period, so about 299 to 359 million years ago, in swamp forests that looked a little like this, although my paleobotany friends tell me that this is much too crowded. But nonetheless, these arborescent seedless vascular plants were the source of much of our contemporary coal. So what was so special about this time in Earth's history that we had all this coal formation? Well, if you want the answer to that question and everything else about coal, you can go to the website of the World Coal Association, where you'll find all the good news about coal. This is a little busy, but this slide from their website presents what I think is sort of the standard model. So they point out that plants trap solar energy in their tissues, and when the plants die, that energy is usually released through the process of decay. The word usually here is important because it implies that decay is something that always happens. It's this uniform process that has always gone on, and this is what usually happens to plants when they die. But under some circumstances in the Carboniferous, because of geological conditions, climate at the time, you had the plant material falling into these swamp forests, getting trapped in anoxic sediments, and then getting buried through tectonic activity, and that led to coal formation. So I don't think there's necessarily anything wrong with this explanation at all, but I'll point out that it is purely explained in terms of abiotic or non-living factors. So this says that climatic factors and tectonic events are solely responsible for coal formation. Now, a different view was advanced by a woman named Jennifer Robinson in 1990, so a long time ago. This is a graph from her publication. The top line that says Sea Org is organic carbon burial, or you can think of that as coal formation, and it's peaking during the Carboniferous period, which I've highlighted in lavender. And what Robinson suggested was that, well, you know, maybe it wasn't just climate and tectonic events, maybe the evolution of organisms had something to do with it. So she talked about plants, which I'm not going to talk about today, but she also focused on the fungal side. Now, in 1990, we didn't have fungal genomes, and we didn't have, you know, lots of molecular clock analyses. We had a few fossils, which are those gray bars on the right, and you can see that there aren't very many during the Carboniferous period, but they become more abundant later. So the lignin-degrading organisms that could have potentially reduced the inputs into those sediments, there's very little fossil evidence for them during the Carboniferous. So Robinson suggested lignin-degrading organisms were rare or absent in the Paleozoic, but diverse and abundant in the Tertiary. So effectively suggesting that maybe the reason that coal accumulated in such great amounts during the Carboniferous is that the fungi that can degrade plant cell walls efficiently, particularly lignin, had not yet evolved or were rare. Okay, so with that background then, here are the questions that I'll address. What's the ancestral nutritional mode in agaricomycetes? What are the genetic bases of these white-brown rot transitions? And how old is white rot? Could the evolution of this mode of decay have contributed to the decline in coal formation at the end of the Carboniferous? And to address this, we've been using genome comparisons. So the genome is the total DNA of an organism, encoding all of its genes, containing all of its genes, encoding all of its proteins, as well as a lot of DNA that doesn't code for proteins. And genome sizes and contents vary widely across life. So some viruses have just thousands of base pairs of DNA with a small number of genes. E. coli, the bacterium, 4.6 million bases, about 4,000 genes. Yeast, the brewers, bakers, yeast, 12 million base pairs, about 6,000 genes. The button mushroom that you buy in the supermarket, about 30 million base pairs, 10,000 genes. That's about ballpark for the organisms I work on. But then you get to mouse, 2.7 billion base pairs, but only 20,000 genes, so not a very efficient use of its DNA if compared to fungi. And a tree, poplar, 480 million bases, 73,000 genes. So fungal genomes are really in the sweet spot for genomics. They're compact genomes, relatively easy to sequence. So they're the Goldilocks genomes. So my work on fungal genomics has been done in partnership with the Department of Energy. In particular, one laboratory in California, the Joint Genome Institute in Walnut Creek. And it might surprise you to know that the Department of Energy is helping me in my work on fungal evolution. I think it's fair to say that reconstructing the evolutionary relationships of fungi is probably not Secretary Perry's top priority. But the DOE is very interested in biofuels and the carbon cycle in general, and these fungi are very important in those processes, and so it's quite logical that they're involved in this work. They've been a superb partner. So I'm going to tell you mainly about one study that we did a long time ago, published actually now in 2012, in which we analyzed the genomes of 22 species of agaricomycetes and 9 others. This is that same tree. Now I've highlighted the places where we've sampled species, showing you their nutritional modes. So 22 is not a huge number, but we tried to sample broadly across the phylogeny, and we also tried to sample diverse nutritional modes. So this is the sampling scheme. And this is another phylogenetic tree drawn in a rectangular format, but it's showing basically the same thing. The red arrow shows you the common ancestor of the agaricomycetes. So we had a lot of things outside of the agaricomycetes. We wanted to put these in kind of a broad context. You'll be seeing this tree in various forms throughout the talk, so let's explain what it represents. So the white boxes in the agaricomycetes are white rot fungi. The brown boxes are brown rot fungi. The one green box is a one ectomycorrhizal fungus. And this is a picture of Dimitri Flutis, a former PhD student who really led the charge on this in my lab. So we inferred the phylogenetic tree, and we found 4 brown rot lineages nested within the white rot lineage, suggesting that white rot is ancestral. So I'm an evolutionary biologist, so this tree on its own for me is a win. This is great to have this resolved. But we also wanted to understand the evolution of decay modes. So we had to look at the genes that encode all of these enzymes. So we looked at all of these gene families, and we're going to talk specifically about the ones that are boxed in white. Okay? So now here's a horrible table. The rows are species. The columns are gene families. On top, the big white box, WR, are the white rot species. Below that, BR, the brown rot species. The colors in the cells, it's a heat map. Warmer colors, higher numbers. Blue means 0. Deepest red is highest numbers, okay? And so if you just kind of blur your eyes, I think you can see that the white rot species have more copies of these decay-related genes than the brown rot species. White rot species, on average, have 142 gene copies in 25 different families from this set we looked at. Brown rot have 74 copies on average in 18 families. So a very much reduced complement of decay enzymes in brown rot. And this is kind of consistent with the idea that brown rot, having evolved this really cool mechanism of using hydroxyl radical to initiate decay non-enzymatically, has been able to cast off some of these enzymes. So we're going to focus just on a few. First, the class II peroxidases, which I've highlighted with the red arrow because we think these are the primary lignin-degrading enzymes. So here's this tree again, which you're familiar with. The bars on the right show the gene copies for class II peroxidases. White rot species on average have 14 copies. Brown rot species have 0 or 1. And when they have 1, we think, based on protein sequence comparisons, that it's probably not a lignin-degrading enzyme. But 0 or 1, whereas white rot species have on average 14. So this is really consistent with the idea that class II peroxidases are important in white rot. These next 3 are enzymes like cellobiohydrolases and others that are involved in attack of crystalline cellulose, which is itself a very recalcitrant material. And it's the same story. So now we're just looking at the agaricomycete portion of the tree. Same thing, expansions in white rot lineages, contractions in brown rot lineages. So the brown rot fungi have fewer enzymes that attack cellulose. Okay, so I want to come back to class II peroxidases here. And what we'd like to do is, we'd like to do more than just have bar graphs, bars at the ends of the tips of the tree. We'd like to actually reconstruct changes in copy number through evolution. As gene families evolve, the gene copies duplicate. Some will duplicate, some will be lost. So the number of copies in a gene family will expand and contract. So to reconstruct that pattern, we actually use the phylogeny not of the species alone, but also of the genes themselves. So this is a complicated tree in which the tips are not species, but they're individual genes. They're individual class II peroxidase genes. And we can take this gene phylogeny and we can fold it onto the species phylogeny to try to reconstruct changes in copy number. This is called gene tree, species tree reconciliation. We can also use this gene phylogeny to understand changes in gene function, but that's a different story. Same tree, only now with colored circles at the nodes that represent copy numbers of these class II peroxidase genes. Gray means 0. White means 1, warmer, darker colors, higher numbers. So we use 3 different methods because these analyses have a lot of slop in them. And they all agree in the basic parameters. So there are some class II peroxidases in the ascomycetes, which are distantly related to agaricomycetes out here. So we have no reason to think that the genes could have hopped between lineages. So what we infer is that 1 copy was retained in the ancestor, the deep backbone leading up to the agaricomycetes, and then initial duplications around the origin of the agaricomycetes with expansions to white rot lineages and contractions to brown rot lineages. And I realize that that's a little hard to see. Potentially this is a messy figure, so I'm going to show you exactly the same results drawn a little differently. Okay? So these are the same trees now stretched out, and the vertical height of the nodes in the tree correspond to copy numbers. So you can see that there's an initial increase around the origin of the Agaricomycetes, expansions to the white rot lineages, and contractions to the brown rot lineages with every method that we use, although sometimes the magnitude of the changes varies. Okay? So we think that this common ancestor of the Agaricomycetes could have been the first white rot fungus, because this is where we first see this class II peroxidase gene family starting to expand. And we'd really like to know how old that organism is. When did it live? So to do that, we have to use fossils. And there's a common myth that there are no fungal fossils, or they're exceedingly rare. And we don't have as many fungal fossils as we do for other groups, but we've got plenty of fungal fossils. Here's a fossil of an organism called Paleopyrenomycetes that lived in the Devonian about 400 million years ago on the left, and on the right, a modern plant pathogenic fungus that looks very similar, at least to me. Here on the left are some fossil mycorrhizal root tips, so a symbiosis between a fungus and a plant, in this case a pine, from the Eocene, about 50 million years old, looks an awful lot like extant mycorrhizae formed by mushrooms in a genus called Suillus that are associated with pines. And we don't have a ton of mushroom fossils. Mushrooms are notoriously ephemeral, but we have a few. This is one that I actually described while I was a postdoc here at Harvard a very long time ago. This is Archaeomarasmius from the Cretaceous period. It's about 94 million years old. And this is from amber from New Jersey, of all places. So this is Archaeomarasmius legati from New Jersey. And we even have some fossil rotten wood. So this excites me. Fossil rotten wood excites me. From the Permian, about 250 million years, and the Triassic, about 225 million years. So these fossils are really exciting because they give us a rich sense of what fungi might have been present in these ancient ecosystems, and that's great on its own. But what's really powerful is that we can use those fossils to constrain the ages of certain nodes on the phylogenetic tree, and then estimate the ages of all the nodes on the tree, albeit with error. This is called molecular clock dating. So we did a molecular clock dating analysis. So same tree with the colored circles at the nodes, but now the lengths of the branches are proportional to time, and the geologic time scale is on the bottom, that sort of tropical lifesavers bar at the bottom there. So molecular clock analyses suggest that the ancestor of the Agaricomycetes existed about 290 million years ago, right around the end of the Permo-Carboniferous, give or take 70 million years. There's some slop in these analyses. And these are the fossils we used for calibration. And this is consistent with these white rot fossils that are a little bit younger and which we didn't use for calibration. So we feel good about that. And this is really consistent with the Robinson hypothesis, right? So we felt really good about that. But of course, there's huge amounts of slop in these age estimates. Our analysis says nothing about plants. It says nothing about impact of tectonics, climate, et cetera. But it's consistent with Robinson's view. So we wrote a very nuanced conclusion, which is here. This is the kind of slide they tell you never to show your audience, so I'm showing it to you. And what we said, we were dancing around obviously, but we talked about the standard explanation that decay was inhibited in anoxic sediments. And then we said, our results are consistent with a complementary hypothesis, which suggests that the decline in carbon burial was caused, at least in part, by the evolution of lignin decay capability. So we thought we were covering our bases and being appropriately cautious. But this was published in Science, and things that are in Science get into the media. And it's not like I have reporters knocking at my door all the time, believe me. But for this story, I had some phone calls. And this person in Scientific American, I think, did a really nice job with his article. It was nuanced and complete. But the headline, white rot fungi slowed coal formation, not exactly what I would have liked. I would have preferred something like, the evolution of white rot fungi may have contributed to the decline in carbon sequestration, but there's considerable error in molecular clock estimates, and besides, this says nothing about tectonics and plant evolution. For some reason, they didn't go for that. But it's the reporter you don't speak to that gets you in trouble, so This blogger said why there's no new coal when reserves run out and how that could help biofuels. Coal-killing fungus could boost biofuel production. And my all-time favorite, mushroom culprit for coal shortage, which is just, it's A, it's just dead wrong, there's no coal shortage, and B, it's a blame the fungus strategy, which offends me. So I'm showing these in part just to be funny, and you're laughing, so thank you. But also to make a very serious point, which is that it's important for us scientists to engage with the media and the general public to explain our science if we want it to be understood and supported. And I guess that's part of the reason why I'm here today. So here's a headline I take much more seriously. This was published in PNAS, and these authors said delayed fungal evolution did not cause the Paleozoic peak in coal production. The Carboniferous Permian peak and subsequent decline likely reflects a unique combination of tectonics and climate, with the particular details of the evolution of plant and fungal community composition bearing no direct relevance. You know, we scientists are argumentative types, and we're always trying to prove each other wrong, which just goes with the territory, and it's a good thing, right? So this is a superb paper. And they make several core arguments. I obviously can't go through this in detail. You wouldn't want me to. But these are the arguments. Lignin decay had to occur before the end of the Carboniferous. I'm summarizing a lot in this paper. Coal formation continued after the end of the Carboniferous. Coal-age plants probably didn't actually make very much lignin, and so if that's a major precursor of coal, then why would the evolution of white rot impact coal formation? And changes in climate and geography are sufficient to explain the changes in carbon sequestration patterns. And all of this may be true, okay? But in response, I mean, honestly, all of it may be true. But in response, I'd just like to make 3 points. The first is that we were not proposing a model in which there was no lignin decay before the evolution of white rot, or no coal formation after the evolution of white rot. Well, I think it is reasonable to suggest that the evolution of white rot could have a modulating effect on carbon sequestration. That's a more nuanced model. The second general point is that, you know, this abiotic hypothesis and the Robinson hypothesis, they're not mutually exclusive. So this is another false dichotomy, right? These are not necessarily mutually exclusive views. And here again, I wanted to show you some of the ways that this research was reported. And I don't mean to denigrate bloggers or journalists. There's enough of that sort of thing going on these days already. But I do want to point out that I think that media people and bloggers, they like controversy. And they like black and white alternatives. And reality is often much more complicated than that. And that's where it's up to us scientists to really lay out our arguments. Anyway, this person said, you know, why was most of Earth's coal made all at once? It wasn't because fungi couldn't break down early trees. This one basically says, and it turns out to be wrong. That's all you need to read here. And then this one, how we got coal, new study fuels the debate, right? Anyway, this is what we're up against. So I said there were 3 points I wanted to make, and the third point is that it's not just about lignin. And I concede that we overemphasize lignin in our paper, partly because in science you only get, 100 words to write your paper. But it's about a lot more than lignin. And here I want to talk about, very briefly, some recent work, just a couple slides. So this is a figure from this JGI microcosm website. So this is the Joint Genome Institute of the Department of Energy. They present online a phylogenetic tree with all of their genomes. So when we did our analysis, we looked at 31 genomes, including 22 from the Agaricomycotina, which is the group shown here. They're highlighted in red. I think you can get a sense of where I'm going with this. There were 165 genomes in 2016. I keep having to remake this slide, but I'm sorry, I didn't do that for you. So now there are 270 genomes in 2018 just of Agaricomycotina, and there are about 1,000 fungal genomes just in the JGI portal. So this data set is just exploding. So with a colleague, postdoc, Laszlo Nagy, we looked at an additional group of genomes, so 62 total, 51 Agaricomycotina, especially emphasizing the early branching groups. And I just want to show you 2 slides to give you a flavor of the results. So on the left is the class II peroxidase reconstruction on the organismal phylogeny. Here, the shading of the branches indicates copy number. So darker shading, higher copy numbers. White is 1, and the dashed lines are 0. And what you can see here is that the ancestor of the Agaricomycetes is actually down here. And we now think that the very earliest groups of Agaricomycetes actually didn't have class II peroxidases. They started to diversify a little later. And we know that now because we've sampled so much more. But other gene families did diversify early, including some of these cellulose-degrading enzymes. So they started to diversify before the class II peroxidases. And then a gene family that's obscure to people who are not enzymologists. These are called cytochrome P450s, very exciting enzymes that can we think they can detoxify lignin breakdown products and other nasties in the environment. They also diversified early in the evolution of Agaricomycetes. So it's a more complex picture. It's not just that class II peroxidases appeared and bam, you had white rot. OK, I'm going to recap because I think we're starting to get close. So white rot evolved through a stepwise general elaboration of the decay apparatus. That's these nodes that are highlighted with the dark circles, including the lignin-degrading enzymes. And this all began early in the evolution of the Agaricomycetes and was retained in these backbone lineages. Class II peroxidases and other decay enzymes continued to diversify in parallel and to expand in white rot lineages and to contract in parallel in the brown rot lineages. And the timing of when this all happened is consistent with this Robinson hypothesis, but these molecular clock estimates have large amounts of error. And they're ultimately dependent on a relatively small number of fossils. We could find one new mushroom could be found in the Rhynie chert, and all of these nodes would get pushed way back. So we do the best with the data we have at hand. OK, I want to conclude with just a couple very general comments. I may not have convinced you that the evolution of white rot caused the end or the decline in coal sequestration at the end of the Carboniferous. I'm not sure I believe it entirely myself either. But I hope I've convinced you that fungi do tremendously important things in the environment. And they've been doing those things for a very, very long time, but not forever. Because fungi are living things. They had to evolve at some time. Nonetheless, when we tell the story of the history of life, fungi are frequently absent. This is a figure from a textbook that we use at Clark. And the title is The Geologic Timescale Showing the Major Events in the History of Life on Earth. There's some prokaryotes, some protists, one plant. But most of the major events in the history of life appear to involve animals. I have nothing against animals. But I would say that the evolution of white rot or wood, those were major events. Okay? But we have, I think, we have a fungal blind spot. And this irks me as a mycologist. And it doesn't help my students get jobs, let me tell you that. But more seriously, I think this fungal blind spot really does limit our ability to understand the functioning and the evolution of ancient ecosystems. And we do have fossils of fungi, and we have excellent phylogenies. We can make inferences about what fungi were present in many different ancient habitats. And once we recognize that they were there, we can ask what they might have been doing. And a lot of that is inferential, right? That's what science is. We make inferences. If you're willing to accept that there were stripes on these dinosaurs, you should demand to see mushrooms on these logs. Thank you.